A single ejaculation of 2 to 3 milliliters of semen contains roughly 100 million sperm per milliliter. This massive verification. Specific surface proteins on both gametes must align and recognize each other for the process to continue. Upon initial contact, the head of the sperm undergoes the acrosome reaction and then releases a specific enzymatic payload. This enzyme, hyaluronidase, chemically dissolves the corona radiata to clear a path through the cellular layer. Specialized structure called the fertilization cone. This cone pulls the sperm inward with a sudden mechanical force. Once inside the egg, the sperm undergoes a precise rotation of 180 degrees. This physical entry acts as a trigger for the egg, prompting it to resume and complete its second maturation division, meiosis II. The breach effectively metabolic shifts begin with a rapid increase in oxygen permeability. This is followed by a sudden spike in hydrogen ions flooding the cytoplasm. This surge in hydrogen ions activates proteolytic enzymes that remain isolated. Once inside the cytoplasm, the majority of the sperm's physical structure is rapidly broken down. Only 3 specific components of the male gamete survive this process. The first is the sperm nucleus, the second is the centrioles, and the third is the mitochondria. The sperm cell disintegrates in the cytoplasm after depositing these 3 specific components. Simultaneously, the male centrioles follow a different path. These centrioles separate and migrate to opposite poles of the egg cell. This polar migration provides the structural scaffolding necessary for future cellular division. Through this process, the male gamete. In the final stage, the sperm nucleus moves toward the egg nucleus. The proteolytic enzymes activated earlier have prepared the nuclear envelopes for contact. This movement leads to syngamy, the permanent physical fusion of the 2 nuclei. This fusion completes the sequence, resulting in the formation of the zygote. The integration of these gametes through specific biochemical and physical stages produces a biological entity prepared for its initial cleavage.